Whole genome sequencing is a powerful tool that offers a comprehensive view of the genetic landscape of cancer. Recently published in Nature Medicine in January 2024, has demonstrated the immense potential of whole genome sequencing in tailoring cancer diagnosis and treatment to individual patients. This study is a big step forward in genomic medicine. It shows how, when we combine long-term clinical data with genetic information, it can predict outcomes and customize treatments, which allows healthcare professionals to make better decisions. Now, imagine you are a super efficient librarian with an incredible speed reading ability. Your job is to read through every single page of every single book, noting down each letter in the exact order as it appears. This is essentially what whole genome sequencing does. Hello everyone, welcome back to another episode of Drug Diaries, where we demystify the wonders of modern science. I'm your host Kaushik Trivedi, and today we have an exciting episode lined up for you. We are diving into the world of whole genome sequencing and its groundbreaking impact on cancer diagnosis and treatment. As always, I'm going to start with the big news for our today's episode and then get into the nitty-gritties of whole genome sequencing using an interesting analogy. So let's begin with the big news from our today's episode. A landmark national study recently published in Nature Medicine in January 2024 has demonstrated the immense potential of whole genome sequencing in tailoring cancer diagnosis and treatment to individual patients. This research highlights how combining whole genome sequencing with clinical data can significantly improve precision cancer care. The study was spearheaded by the Genomics England, NHS England, Queen Mary University of London, Guy's and St Thomas's NHS Foundation Trust, and the University of Westminster. These institutions came together and analyzed data from over 13,000 cancer patients covering more than 30 types of solid tumors. The study data used in this project was from 100,000 Genome Project. This was introduced in 2012 with the aim of sequencing 100,000 genomes from patients with cancer and rare disease to drive the integration of genomics into UK healthcare. This study is a big step forward in genomic medicine. It shows how, when we combine long-term clinical data with genetic information, it can predict outcomes and customize treatments, which allows healthcare professionals to make better decisions. Now, let's take a look at the whole genome sequencing itself. What is whole genome sequencing and how does it work? So, whole genome sequencing is basically a comprehensive method for analyzing the entire genetic code of an organism. Unlike targeted approaches that focus on specific genes or regions, whole genome sequencing examines the complete DNA sequence, providing a full picture of the genome. Now, in order to simplify the concept of whole genome sequencing, I would like you to imagine the genome as an enormous library containing all the information needed to build and operate a living organism. Whole genome sequencing is like taking on the ambitious task of reading every single book in that library, cover to cover, and cataloging all the information. And here's how we can break this down. The library, which is a metaphor for genome, is made up of 23 pairs of bookshelves, which is the metaphor for chromosomes. On these shelves sit about 20 to 25,000 books, which are your genes. Now, each book is written using an alphabet of just 4 letters, A, C, G, and T, representing the 4 DNA bases, mainly adenosine, cytosine, guanine, and thymine. Now, imagine you are a super efficient librarian with an incredible speed reading ability. Your job is to read through every single page of every single book, noting down each letter in the exact order as it appears. This is essentially what whole genome sequencing does, and it determines the precise order of all 3 billion DNA bases in a person's genome. But why go through all this trouble? Just as each book in a library contains valuable information, each gene in our genome holds instructions for making proteins which are crucial for our body's functions. If you are able to read and understand the entire library, you can identify genetic variations that might cause diseases, discover new genes and their functions, understand how different genes interact with each other, and also trace our evolutionary history and relationships with other species. It's like having a complete manual for a human being. And with this knowledge, we can better understand how our bodies work and why certain diseases occur, and potentially develop more effective treatments. The exciting part is that as the sequencing technology improves, it's becoming faster and cheaper to read this library of life. What once took years and billions of dollars can now be done in a matter of days for a fraction of cost. This opens up incredible possibilities for personalized medicine where treatments can be tailored to an individual's unique genetic makeup. So the next time you hear about whole genome sequencing, remember, it's like speed reading through the most fascinating library in existence, the one that contains the blueprint for life itself. This technique usually involves a number of steps. Typically, a blood sample is taken from the patient and then DNA is extracted from the sample and broken into smaller fragments. These fragments are then sequenced and then the sequences are then assembled to reconstruct the entire genome. The assembled genome is then compared to the reference genome to identify any genetic variations that may be causing the disease. Whole genome sequencing has revolutionized cancer care by enabling personalized and precise treatment strategies. And there are several ways it enhances patient care. One is identification of genetic mutations. Whole genome sequencing can detect a wide range of genetic alterations, which includes single nucleotide variants, insertion, deletion, copy number changes, and large structural variants. This comprehensive detection is crucial for understanding the genetic basis of cancer. Another area where whole genome sequencing plays an important role is in the development of personalized treatment plans. By comparing the DNA from a patient's tumor to their healthy tissue, whole genome sequencing can identify specific mutations that are driving the cancer. This information helps doctors tailor treatment to target these mutations, improving the effectiveness of therapies. For example, certain mutations may indicate that the patient will respond better to a specific drug or treatment regimen. Next area is the avoidance of ineffective treatments. So whole genome sequencing can avoid the use of treatments that are not working and thereby reducing the unnecessary side effects. By understanding the genetic profile of the tumor, clinicians can predict which treatments are unlikely to work and focus on more promising options. Third area is the early detection and risk assessment. In some cases, whole genome sequencing can identify inherited genetic variants that increase the cancer risk. This information is valuable not only for the patients but also for their family members who may benefit from early screening and preventative measures. And last but not the least, whole genome sequencing can help improve prognosis and monitoring. It provides detailed insights into genetic changes in cancer cells, which can help predict disease progression and response to treatments. And this allows for more accurate prognosis and better monitoring of the disease over time. So as we are approaching the end of our today's episode, I'll just take a minute to just summarize some of the key points we covered on the topic of whole genome sequencing. So number one, whole genome sequencing is a powerful tool that offers a comprehensive view of the genetic landscape of cancer. Number two, by identifying specific genetic mutations, it enables personalized treatment plans. And three, it avoids ineffective therapies. Four, it provides valuable insights into disease progression and risk. And number five is, as the sequencing technologies continue to advance and become more accessible, whole genome sequencing is poised to play an increasingly central the outcomes for cancer patients across various types of cancer. I hope you found this information helpful. Before we wrap up today's episode, I would like to extend a special invitation to all the innovators and trailblazers in our audience. If you are working on something truly groundbreaking, whether it's in the field of medicine, technology, AI, commercial, patient safety, product engineering, medical, regulatory affairs, or any other domain, and you would like to share your work with a wider audience, we want to hear from you. Drug Diaries as an initiative is committed to shining a spotlight on the incredible work being done by visionaries like you. So if you have a story to tell, an innovation to showcase, or a breakthrough to unveil, don't hesitate to reach out to us. Simply drop me an email at drugdiariespodcast@gmail.com and let's start a conversation about how we can feature your work on an upcoming episode. That's all for today, folks. Thank you for tuning in to Drug Diaries. Until next time, stay curious, stay inspired, and keep changing the world one breakthrough at a time.